Okay, so the last session we looked at the UniProt, and one of the things that I showed was this protein, angiotensin converting enzyme 2, and my point was that it has a structure that has been solved. Okay, and on this, on the right hand side, you'll see the different, well, not is the structure of this same protein that have been deposited on the protein data bank. Okay, let me just take it. You see that they have specific, what we call a PBD entry, specific ID, and the method by which the structure was solved. See stratigraphy. It was a model built computationally. There's also electron microscope as a way to solve the structure as a method. Then you have the resolution of the solved structure. The higher the resolution, the more confident you are that the experiment actually got the coordinates of the amino acid, like I spoke about, the correct coordinates of the amino acid in the final 3D structure. So you want to pick the highest coordinate. The X-ray structures is okay, but modern strategies of using electron microscope, so you can also explore the structure that are built from that. Then the chain that is in the protein, sometimes they might just model or resolve one chain. The chain refers to the fact that, like I said, a protein can have two different 3D structure that comes together in a dimer or tetramer or something like that. So sometimes you might have they are able to resolve both major chains involved in that protein, A and B. Some just did the A part of it. Yeah. So depends on what the experiment that was done and which part of the structure was resolved. Some have everything, different aspects or different chains that constitute that particular protein. All right. But yeah, so you can actually now, and this is the position in terms of the length of the protein, in terms of sequence length, and finally links to where you can get them. So you have the PDB Europe, PDB RSSB, maintaining the US. I think what was there again? Look further. There we go. Research Collaboratory for Structural Bioinformatics. They also maintain a database to store digital representation of 3D model of protein structures. Okay, where are we? Yeah. And there's still PDB Japan and the summary PDB page. So I think I opened one of them. Okay, this one, the PDB sum gives you like a summary page of the entry, and describes. So in this case, the ACE receptor, ACE2, like I said, is the main receptor for SARS-CoV-2. So the structure was actually resolved together with SARS-CoV-2. Okay, so you can see that's the spike protein of the SARS-CoV-2, and that's your ACE2. All right. Some quality checks are done, which I will show you the details when we get to the PDB. So I'm just going to from this page, like I said, you can get straight from the UniProt with a link to PDB. But assume that you went straight to PDB. So let's say you went straight to, so I'm going to go back to Google. If you can just type it straight if you want to. And I'm going to say, if you write PDB, I think in my tab, it takes me to the RCSB PDB homepage, and that's most frequently PDB page that I use. Okay, so you can type it straight here, or you go straight to the home page, and now then on the home page you have the description and some news about molecules of the month, recent submissions, and the rest of them. Okay, but this is where your interest is. So I can type in the angiotensin converting enzyme 2. All right, so it gives me quite a number of options, but I'm just interested in that. I don't want it linked to, yeah. So most of the entries, because of the recent coronavirus, have the ACE linked to the spike protein of the SARS-CoV-2. Okay. Most of them have that. So these are the different entries that have been submitted to put into the bank. Now this one is the native human angiotensin converting enzyme related captosylpeptidase. It's linked to a particular peptidase. Okay, so it might be an interesting one to look. So this one also has an inhibitor attached to it. So let's take a look at this one. There might be some interesting things to see. Okay, so you have a description of the protein itself. It's a hydrolase, Homo sapien, the organism, where it is located, where it was found. It was expressed in this system before the resolution was done. Okay, that's the date it was deposited. Of course, it was checked and curated, released on this particular day. You have the link to the paper of the guys that actually worked on the resolution. All right, so just here, I just want to show you that each resolved structure is analyzed, and there's a quality check done using different parameters, and they are scored based on their performance, based on that particular, on each of those parameters. Here we have something called a pro check on that PDB summary. And one of the things that is there is your Ramachandra plot. We can check that out. It's a way of checking if the amino acid are all in the appropriate position that is allowable based on the bond that they carry or share with other atoms in that protein. And you can just check it out. But here they give this metric a R-free class score, Ramachandra outliers, based on this Ramachandra plot that I showed you, side chain outliers, and the rest of them, and you can go into details, just look at the full report, tells you the quality. All these are metrics that tells you the quality of this structure that you're about to work with. And remember, the resolution is also given. Yeah, the higher the resolution, the better it is. All right. Okay, so a couple of things I just want to show you here. The structure itself, you can click on the 3D viewer. So the PDB database also has a visualization tool. allows you to visualize this protein. So if you click on that, it takes you to a visualization tool. I think this is powered by NGL that power this visualization, I think. Okay, so yeah, there we go. So with this, you can move the protein around and view the different domains of that protein. So that's your ACE tool, and that is the protein that was attached to it, that was bound to it. Maybe there's some people are trying to see if this peptide can inhibit this particular protein. Okay, I'll just go back again. What else I want to show you? The ligand interaction. So there's a ligand bound to this. So let's see how they interact. You can also click on that. Mhm. All right. All right. All right. So this is a ligand that is bound to this protein. And this is you can actually look at the identity of this if you put it. Just you will see the identity just at the bottom there. If I just point my cursor on that, it's a 2-acetamido 2-deoxybeta D glucopyranose. Pyranose is a sugar attached to that protein. So you can zoom in and zoom out. So you can play around with this and just check proteins of your interest, just type. And you can also um, say a class of particular protein. If I say protease present in HIV virus. So it gives me, yeah, a couple of those proteases I can look at. Let me see. This is bound to a particular inhibitor. Just take some time to come up. Here we go. Okay. See the protein with the ligand in it. So that's the ligand. Let's see if we can get the identity of that ligand. Okay, it's not showing here. There are multiple of them here. And if you go closer, you'll see how the ligand is interacting with the residues of the amino acid through these bonds, hydrogen bonds, yeah, that they form with the residues or the protein. All right. You can also download this. Click on that. You can either download the faster sequence, but in our case, we need the 3D model, so you can download the PDB file, or you want to zip the file for or the PDBX, but I would prefer on the platform that we're working, the PDB format should be fine. So you click on that, then you ask you to save it. You click on save. You browse to the folder. I would suggest you have a folder for the training, and also it's folder for each session of the training within that same folder. So in this case, you went to the my downloads. Okay, so please, you can actually move it from here to the specific folder that you want. Okay. I think that's about what I'm going to there's a hands-on exercise that we want you to do. Let's take a look at the PDB and download a particular protein that you're going to use for the docking simulation. First, we're going to just visualize that, then before we do the docking simulation. Okay. So the next session we will be looking at how you can visualize these macromolecules of proteins and how you can manipulate them as required. I'll talk about that.